Good morning, all. Today we want to see how knowledgeable we are in basic microbial genetics. We want to measure our knowledge level in basic microbial genetics. So, if you're asked to explain general principle of microbial genetics, what are you expected to do? You should be able to define what microbial genetics revealed, what observation, the foundation of microbial biology has, what did the subsequent investigation of bacteria revealed, how is bacterial DNA compared to that of human. Another question is, if you are asked to summarize DNA replication, what do you do? No matter the story that you prepare to tell, don't forget to include when separation of DNA strand, when it takes place, by what led to synthesis of new strand, synthesis of new RNA, what enzymes plays role, what happened, what leads to messenger RNA strand, and proteins. These are things that you must pay attention to. Then if you're asked to differentiate between prokaryotes and eukaryotes, what are the differences? Then if you're asked to explain the steps involved in transformation, I hope you remember, you know what you're expected to do. You should be able to tell us what happened to the donor bacteria when it dies, to release DNA. The DNA, what happened to DNA? It is graded to fragment, then the fragment binds to binding protein on the cell wall. Then a nucleus enzyme called the bound DNA into fragment. The one strand is destroyed while the other penetrates into recipient DNA. And then lastly, we'll have genetic exchange. Then if you're asked to discuss method of gene transfer mediated by bacteriophage, you have to first of all mention the name of the gene transfer, explain what bacteriophages are and what happened during viral replication, explain lytic and lysogenic phase, and how gene transfer occurs in these phases. Again, if you are asked to explain bacterial gene transfer from living donor to recipient, you should be able to mention what type of gene transfer is that and explain what plasmid transfer in gram-negative bacteria looks like. Then if you are asked to define what plasmids are, you should be able to know what are plasmid, types of plasmid, significance of plasmid. Again, what are transposable genetic elements, what they are and what are their properties. What are types of transposable genetic elements? Explain the relationship between mutagenesis and mutation. In my last class here, I explained it in a very simple term. What are the 3 reasons why mutagenesis occurs? I also mentioned that in our last class here. Then explain endogenous mechanisms of mutagenesis. I also mentioned that in the last presentation here. Then list exogenous mechanisms of mutagenesis. Then if you're asked to define site-directed mutagenesis and outline 3 steps involved, I hope you do that perfectly. Highlight clinical significance of mutagenesis. Of course, if you're able to attend to these questions, then there is no doubt that you really have good basic knowledge of microbial genetics. Thank you and remain blessed. See you next time at this place. Make sure that you like, you subscribe, you share. Thank you.